Sure. So my work is focused on the blood-brain barrier. And what the blood-brain barrier really means is just a term used to describe the unique properties of blood vessels that vascularize the central nervous system. And what it means is that blood vessels in our central nervous system have different properties than blood vessels in other tissues. And these properties allow these blood vessels to tightly regulate the environment of the central nervous system, which is both important for nervous system function and also to protect it from injury and disease. And so my work really focuses on kind of three main questions. One is, how is the blood-brain barrier formed during development? So what are the cellular molecular signals that tell the endothelial cells to form this barrier? What goes wrong during injury and diseases, such as Alzheimer's disease? And then third is, how is the blood-brain barrier dynamic? How does it respond to changes in brain activity? And then how do those changes respond, how do those changes affect the function of the brain? And that's actually what I'm going to talk about tomorrow, is work that we've been doing trying to understand how dynamic the blood-brain barrier, how has it changed, and then how do those changes affect the brain, and how could kind of those dynamic changes then, if you have errors in them, how could that potentially lead to diseases such as Alzheimer's disease? We wanted to understand how the blood-brain barrier is dynamic, and basically the reason is that people previously have been studying the blood-brain barrier as a wall, just a static wall that's there to protect the brain. But we know it's not. We know it's all sorts of properties. It's structural, physical, transport, metabolomic, signaling, all these properties that allow blood vessels to regulate the environment. And so what we wanted to know is, how dynamic are those properties, and how do they change in response to both stimuli coming from the brain and from the environment? And so I'll talk about work that we've done tomorrow, showing that when we manipulate neural activity, you actually see profound changes in the blood-brain barrier. And I'll talk about one of those changes, and that's this efflux transport system. So this efflux transport system is incredibly important to extrude potential toxins from the brain. And what we found is that neural activity can actually control the amount of this efflux system in the brain. With the more neural activity, the less of this kind of waste removal system. The less neural activity, the more. And we further showed that there was kind of a circadian a diurnal oscillation to this efflux, and that neuronal activity could use circadian clock genes to regulate the amount of this efflux. And so why this efflux is important, especially in the context of Alzheimer's disease, it's been shown to remove A-beta from the brain. And so what this tells us is that if you have, since this efflux is dynamically regulated, if you get errors in this, potentially if you have too much neural activity, then potentially you would lose this efflux and you would lose the ability to remove A-beta from the brain, which could potentially exacerbate Alzheimer's disease.